Thanks, Brett, for that really kind introduction. There's some feedback. So the title of my talk is Using Population Genomics to Gain Insights about the Evolution of Peromyscus on Islands. And so islands have held very prominent positions in evolutionary biology because they serve as these so-called natural laboratories for us to study processes like speciation, adaptation, and the repeatability of the evolutionary process. And so, like many others, our lab is fascinated by evolutionary outcomes on islands. And like Brett said, our lab is motivated by, or a lot of the research in the lab at least, is motivated by this ecogeographical pattern called the island rule, which describes this tendency for small species to get big on islands and big species to get small. And this figure from a recent meta-analysis, I think, nicely illustrates how this relationship between island and mainland body size holds across diverse taxonomic groups. And so my dissertation research involves characterizing two island-mainland systems of Peromyscus mice, featured here. One concerns Peromyscus leucopus, the white-footed mouse, in the Boston Harbor. And this map shows the locations, the two island locations, Pedix and Bumpkin, and the one mainland location from which we've sequenced whole genomes of wild-caught mice, indicated by these sample numbers. The second system concerns Peromyscus maniculatus, which is the deer mouse, and this map shows where we've sampled mice from these two islands, Saturna and Pender, and one mainland location called Maple Ridge. And so the reason that we've constructed or sequenced all of these mice and constructed these population genomic data sets is because island mice in both of these systems exhibit increased body sizes consistent with the island rule. So our first step in characterizing these populations is to reconstruct their demographic history. And the demographic history provides really important context for understanding how these phenotypic differences that we can observe evolved. So we can ask questions like, when did island and mainland populations diverge? And this can help us distinguish between different scenarios of island colonization, whether that's by vicariance or by natural dispersal events or by human-mediated introductions. We can also ask how population sizes have changed through time. And this tells us something about the efficacy of natural selection in these populations. And we can also ask to what extent there's been migration between populations. And this is important for understanding how those phenotypic differences arise and are maintained. So to estimate these features of the demographic history, we use an approach based on the site frequency spectrum with a little toy frequency spectrum here. And the site frequency spectrum is a really important distribution in population genetics. And it's a really straightforward way of summarizing sequence data. So it's simply a histogram that counts the number of SNPs or mutations that are observed in each frequency bin in a sample of individuals drawn from a broader population. And despite its apparent simplicity, the site frequency spectrum is actually a very useful readout of population history because it summarizes the underlying genealogical relationships in our sample. And this figure shows how different population size histories can distort, and those are represented by the yellow boxes, can distort this underlying genealogy and, as a consequence, the site frequency spectrum that we can observe. And so we used an approach or software called Moments, which in a way leverages this logic to conduct maximum likelihood inference of demographic parameters. And then we applied this approach to those population genomic data sets that we constructed for both island and mainland systems. And I'm going to skip over a lot of the methodological details and get right to what we learned about the demographic histories. And I'll start with the Boston Harbor system. So in this system, we fit demographic models to each pair of populations. So shown on the left is our mainland, World's End, and Bumpkin Island comparison. All of these populations have very funny names. In the middle is our comparison between the two islands, and on the far right is our comparison between the other island and the mainland population. And so these diagrams illustrate our best fitting demographic model, which involves a simple split of our contemporary populations from some shared ancestor. And what's notable is that in each of these three cases, we find that our best fitting model does not involve migration either between island and mainland or between the island populations. So the estimates for each of these parameters are given in bold. And the general takeaway is that all of these pairwise contrasts are telling a similar story. So we find the smallest effective population sizes in our Bumpkin Island sample, or for our Bumpkin Island population, followed by our Pedix Island population. And we estimate the largest effective population sizes for our mainland World's End population. And what's interesting is that features of this demographic history appear to be congruent with the geological history of the region. So if you take a look at the split times that we estimated, they all are around 7,000 generations ago, which, depending on what we take to be the generation time of Leucopus, is somewhere between 3,000 and 4,000 years ago. And this corresponds roughly to when the islands themselves became isolated from the mainland due to rises in sea level following the last glacial maximum. So it's really nice to see that type of agreement. And it also suggests that, indeed, the establishment of these island populations probably follows this vicariance model. Okay, so we can do the same sort of exercise for our other data set of these Gulf Island populations. In this case, we decided to fit a 3-population demographic model to all of our to all 3 samples simultaneously. So it's a little bit more complicated, but I'll break it down. So this joint model involves 2 historical epochs. One that involves the split between an ancestral island population and an ancestral mainland population from some ancestral population, and migration between the 2 of these. And then the more recent epoch describes the divergence of all of our contemporary populations from their respective ancestors. And unlike what we found in the Boston Harbor system, we actually, our best fitting model actually does involve migration between our 2 island populations, but not between the islands and the mainland. Here are the estimates for each of these parameters. And similar to the Boston Harbor populations, we see that the effective population sizes that we estimate for both Saturna and Pender Island are smaller than the effective population size we estimate for the mainland population. And if we look at the timing of this second epoch, it also corresponds, or it's within the bounds of what we would expect for the formation of these islands in this system. But we were really surprised by the very old divergence times that are implied by this earlier epoch, which suggests that the ancestral island and ancestral mainland population diverged over 2 million generations ago. And this way predates the sort of Pleistocene glacial activity that is responsible for contemporary land areas. And so that was very surprising. So in some ways it's congruent with the geological history, but there are still some mysteries here. Okay, so we are now armed with these 2 working models of demographic history in both of these systems. And now I'd like to shift gears for the remainder of my talk and instead focus on patterns in each of these data sets that's not captured by these histories that we inferred. So I'm going to discuss what we think are the causes of these patterns and how they contribute to our understanding of evolution in both of these systems. So I call this a deep dive into 2 genomic patterns. And the first concerns rare variants in our Boston Harbor populations. And so when I mean rare variants, I'm referring to singletons. And if we return to our little toy site frequency spectrum, those are referring to SNPs that are observed only once in the sample. So for example, if I sampled 4 mice, those mice are diploid. A singleton represents the case where one of the chromosomes of one of those mice carries the variant in question. And we expect that these should be distributed, not necessarily randomly, but throughout the genome. And indeed, if we look at where these singleton variants fall in our mainland sample, it corresponds to that expectation. So these plots are very messy, so I'm going to break it down. The x-axis plots physical position along chromosome 1 in this case. And each of these rows represents these singleton variants that are contributed by a specific mainland mouse. So all of those mice have IDs in different colors. So for example, this smattering of blue tick marks represents singletons contributed by W2. And each tick mark denotes the location of one of these singleton variants along chromosome 1. And so the distribution is somewhat patchy. We can see that there are instances where we don't observe singletons in any sample, but this is largely driven by the way that we filtered and masked particular regions of the genome. So for the most part, this genomic distribution of singletons looks normal in the mainland sample. It does not look normal in either of our island samples. So what's shown in this middle facet is the genomic or the distribution of singletons along chromosome 1 in our bumpkin island sample. So as before, each of these rows represents a different mouse, and the tick marks the location of singletons. And the bottom panel represents that same information for our Pedix island sample of mice. And in contrast to what we see in the mainland sample, singletons are concentrated into these very defined tracks where one mouse is contributing all of the singletons. And this is very unusual. And what's even more unusual is that if we take the singletons within these tracks, we find that in some cases they're shared with the other island and the mainland population. So that sharing is suggesting that this pattern is unlikely to be caused by genotyping error. And if we return to our map of the Boston Harbor archipelago, there are a lot of islands in this system and potentially a lot of opportunities for the movement of mice. And so it's possible that this pattern reflects some form of migration. And our rationale, we can walk through. So if we consider that our contemporary populations, which are denoted by these circles, are samples from some ancestral pool of polymorphism, represented by these colorful dots, over time, because of genetic drift, the polymorphisms in these initial samples in these initial populations become lost or fixed due to drift, and it increases the uniqueness of each of these populations on the left and the right. And so when there is migration, for example, from our right circle into our left circle, much of the variation that's carried on this migrant haplotype, which is denoted by this second chromosome, is going to appear or manifest as singletons in our recipient population. So the caveats to this little verbal model are that it requires that there's sufficient divergence between our two contemporary populations, and it requires that migration is rare enough such that the variants that are carried on this migrant haplotype always look new in the recipient population. Okay, so to investigate whether this is a likely cause of the pattern, we returned to those demographic models we had constructed for both the Pedix Island and the Bumpkin Island populations. And we augmented those existing models with migration from some unsampled so-called ghost population. And this occurs following the split of all three populations, island, ghost, and mainland, from their shared ancestor. And essentially, this is meant to represent migration within the archipelago that's moving around ancestral polymorphism, but we haven't sampled the source of those migrants. And so we conducted simulations under this model of ghost migration, and we tested a range of different migration rates. And we also varied the time at which migration begins following the split. So on one extreme, migration can occur over the entirety of the split, while on the other extreme, migration can begin only very recently. And then we asked if any of these simulated regimes were capable of recovering this pattern of singleton clustering that we observed in both of the island samples. And the tentative answer is yes, maybe, at least in a very qualitative sense. So what these facets are showing are simulated chromosomes for bumpkin at the top and for pedix at the bottom. And these are arranged as before, where each row represents a different individual, and these tick marks are singletons. And the top facets in both cases, those chromosomes were simulated under a scenario of weak migration that's been occurring for a long period of time. And the bottom facets in both cases are chromosomes that represent strong migration that's only just begun occurring very recently. And in both cases, we see this smattering of background singletons, but we also see these defined tracks that are reminiscent of what we observed in our real data. So this is a very preliminary characterization of the pattern, but it could help us to revise our understanding about levels of connectivity between the populations in this system. And what's useful about this particular signature is that it doesn't actually require that we've identified the source population of these migrants. And so it could be used to test for migration in fragmented systems where it's not necessarily feasible to sample every population. But that's going to require future work. In the meantime, I'm going to shift gears to our other data set representing the Gulf Islands populations. And I'm going to talk about patterns that we observed in allele frequencies and linkage disequilibrium. So this story also begins with site frequency spectra, one for each of our Gulf Islands populations. So these are just empirical examples of that toy SFS I showed earlier. On the left is the Saturna Island site frequency spectrum. In the middle, the Pender Island SFS, and on the right, the mainland Maple Ridge SFS. And what we noticed were these strange bumps in the genome-wide site frequency spectrum for each population. And these bumps depart from our expectation that under most demographic histories, we should expect the height of these bins to decline in a monotonic fashion. And what we found was that these bumps are actually being driven by specific chromosomes. So down here, I've plotted the Saturna Island site frequency spectrum, 4 SNPs drawn from either chromosome 21 here, 7, 15, 14, or 22. And what you can see is that these minor distortions in the genome-wide site frequency spectrum are actually being driven by major chromosome-specific distortions in allele frequencies. And we found that these same 5 chromosomes were also driving these bumps in the Pender Island sample or site frequency spectrum and in the mainland Maple Ridge site frequency spectrum. And these same 5 chromosomes also show distortions in the decay of linkage disequilibrium, or LD. So to measure linkage disequilibrium, we use R², which represents the correlation in genotypes between 2 loci. And if we consider loci that are separated by a large distance, then the increased opportunity for recombination events should break down this correlation. And so we expect that R² is going to be lower for loci separated by greater distances. And that's what causes, or that's what produces this characteristic LD decay curve. However, if we plot the same curve for those 5 chromosomes for each of our populations, which are denoted by the different colors, what we see is that there are these distances over which LD is much more elevated than we would expect. So to figure out what's going on here, we took a closer look at the genotypes of SNPs that are contributing to each of these elevated allele frequency bins. And so, for example, we can take SNPs with a minor allele count of 17, and we can plot each mouse's genotype at these chromosome 15 SNPs. And that looks something like this, which is a bit of a messy plot, but I'll break it down. So the x-axis represents the physical position along chromosome 15. Each of these rows represents the genotypes for a single mouse, specifically at SNPs that have this minor allele frequency. And then the color of each tick mark denotes what that individual's genotype is at that site. So they're either heterozygous, homozygous alternate, or homozygous reference. And what you can see is that there's this extreme genotype continuity over what is almost half of the chromosome. So for example, the pink bars at the top represent individuals that are heterozygous for these bin17 SNPs over what looks like a little more than half the chromosome. And same patterns for the green bars and for the blue bars. We find this same pattern in our for Pender and for mainland Maple Ridge. And if we do this same exercise for those other chromosomes that have these allele frequency distortions, we get this same type of plot. And so what we think is actually going on here is that these genotypes reflect large-scale inversion polymorphisms, and that the genotypes of these mice are actually telling us something about the arrangement of these inversions in each of the mice. So an inversion, like it sounds, is a section of chromosome that has an opposite orientation to whatever we consider the standard orientation, and they can arise for a variety of reasons. And the reason, or the, I guess, support we have of this hypothesis comes from the overlap between the genomic intervals that are showing this pattern in our data and previously identified inversion polymorphisms in Peromyscus maniculatus. So this figure comes from a recent study that used a combination of population genomic data and long-read sequencing to basically catalog inversion polymorphisms in maniculatus across its geographic range in North America. And what they found were 21 very large inversion polymorphisms, and this plot shows where they fall along the 23 autosomes. So they're represented by these gray boxes on each of the chromosomes. And the yellow boxes represent genomic intervals that exhibit this pattern in our data that overlap with these known inversion polymorphisms. So what's interesting is that this study documented substantial geographic variability in the presence of these inversions. So this figure is complicated, but each of these letters represents a different population they sampled. And those are also denoted here. And these pie charts represent the frequencies of different inversion genotypes in each of those populations for all the 21 inversions that they identified. And essentially, the takeaway here is that different populations tend to segregate different collections of inversions. And so it's really surprising that we see this congruency among the Gulf Islands populations with respect to which inversions they're polymorphic for. And so if we go back to the demographic history that we've inferred for these populations, it actually constrains different scenarios for how these inversions emerged. So in order for the same inversions to be present in all of our contemporary populations, it's possible that they were present in the ultimate ancestral populations, or it's possible that they were introduced via migration during this period of gene flow between the island and the mainland ancestor. Or it's possible, but probably much less likely, that they have independent recent origins in all of our contemporary populations. And these first two scenarios are interesting because they imply that these inversions might actually be quite a bit older than we would expect for neutrally evolving loci. And so it's possible that the reason we see them in all of our contemporary populations is because they're being maintained by some form of balancing selection. So lots to do here as well. But I think that these two tangents provide a nice segue into the topic of rigor and reproducibility. Hopefully, I've emphasized the importance of following up on unique features of the data. In some cases, they can reveal novel evolutionary insights, and in other cases, they can reflect problems with your data, which no one likes to see. One thing that's helped us distinguish between these two scenarios is a practice our lab has adopted of sequencing technical replicates when we're constructing these population genomic data sets. And so what this means is just taking some mice and sequencing them twice. And this is really useful because by comparing genotypes at these technical replicates, we can identify genotyping errors. And if we can do that, then we can also adjust our filtering practices such that we achieve an optimal balance of sensitivity and specificity. So to summarize, we have useful demographic models for both the Boston Harbor and Gulf Islands populations, and they, in some cases, agree with our expectations based on the geological history of each region. We identified this pattern of rare variance in the Boston Harbor populations, which is maybe suggestive of recent or rare migration. And these distortions in allele frequencies and LD decay in our Gulf Islands populations indicate that they segregate possibly old large-scale inversion polymorphisms. So future work is going to leverage these demographic histories I've inferred as null models to ask questions about purifying and positive selection. And with that, I'd like to thank the lab, both members past and present, my thesis committee, and all of our funding sources. Thank you, guys, and I'll take any questions. Nathaniel.
If the inversions are totally new on the islands, then wouldn't you expect those to have maybe the same location if there's some pattern to where inversions occur, but different SNPs within the inversion relative to what the Huckstera data show? Can you compare the actual SNPs to tell whether these things are ancient or new?
Yeah, so are you envisioning a scenario? Because, I mean, part of the reason these inversions are maybe, or one of the determinants of where these inversions are occurring in the genome has to do with like repetitive DNA content and the propensity for those to generate the inversions. So I guess in that sense, I can see why there would be similarity in their locations. I haven't, I'd like to characterize them in more detail, but yeah, I suppose I could look at like divergence between, because this way of in a way genotyping the inversions lets me like, you know, look at inversion heterozygotes and standard, or inversion homozygotes and standard homozygotes. I can separate out populations of mice that only carry the inversion versus only carry the standard orientation. And in that way, I could compare SNPs and like look at divergence between the inversions in this data set, both between populations in the Gulf Islands, but also between populations that the Hoekstra lab characterized. Yeah.
Awesome talk. When you do the technical replicates, do you have a replicate for each set, for each individual, or do you just keep several to try to catch the instrumental sequencing?
So this is actually something we implemented specifically for the Gulf Islands data set. And in that case, well, you know, in the best case scenario, you can sequence every individual twice. In that case, we chose 4 mice to sequence twice, and we sort of replicated them at the level of library prep. So these are capturing errors introduced both during that process and also during sequencing and then downstream genotyping. But no, we're not doing all of the mice. We're just doing a representative set. And our hope is that, you know, those few individuals can sort of inform our filtering practices.
Do you know what proportion of the, whole genome is error, like based on your 4 replicates?
Oh yeah, so that's something we estimated just by simply comparing genotypes. Like I said, that considers all these possible sources of error in the process. I don't actually remember what that number is, but yes, we can estimate the rate of error and also like say something about like how these erroneous sites are distinct from like sites that are called correctly, basically.
Do you think that the variation would be like in the island populations is leading to like different standing variation in the populations, just generally creating like a more diverse populations of like that species of mouse, or do you think over time these inversions would lead to like a like speciation events because they could no longer recombine?
Well, they certainly probably have a pretty big impact on the recombination landscape because there's so many of them, and because in heterozygous individuals, recombination is going to be suppressed. Yeah, I think it. I don't know if I have a good answer for you, but yeah, I think I'll learn something more by doing comparisons between these inversions in our populations to what's already been characterized. Chris?
Many questions. Try to keep it short. Do you think the mice do compensate for aneuploid loss of offspring, or do you think they suppress recombination and therefore don't need to?
I actually, so a lot of the evidence pre sort of genomic era for these inversions comes from like cytological studies, or I guess cytogenetic studies. And I'm sure they've characterized what's happening in these inversion heterozygotes. But as far as I know, I think recombination is just being suppressed.
When you do sweeps and purifying, are you going to treat the inversion population and the standard population separately for each of those?
Yeah.
Do it for both.
Or just not do it for the inversions because
Don't
really hard to what?
do it for the inversions.
Yeah, well, I mean, it's this there's this conflict because inversions have actually been shown to be associated with differences between forest and prairie ecotypes of maniculatus. So certainly they have consequences for adaptation, but at the same time, it's hard to construct a null model for the dynamics of these inversions because it depends so much on when they were introduced, their sort of frequency trajectory through the population, because that determines how often they're found in a homozygous versus heterozygous state. Like there's just a lot of unknowns there. So I think that I'm probably just going to ignore them for that. But I could, do something simple like an HKA test to see if these inversions truly do depart from like our neutral expectations.
Well, I wasn't thinking the inversions themselves as alleles. I meant within the population, like treating them independent of their fitness effect as an inversion. The effect of variance sweeping within the haplotype class, so the karyotype.
Oh, like within the inverted and
Yes.
yeah. We'd have to talk about how to do that.
We should probably transition to our second topic.
Okay.
Thanks, Ed.
Thank you.